Gunnar, the floor is yours.
Okay, fantastic. Can you guys see my my first slide, or does it say like loading or something?
No, we can see
You can
it.
see it just fine.
Great, fantastic. All right, I'll work on the assumption that everything is good unless I hear otherwise. Let me know if you have any questions. I do a little bit of a warm up here because this is a very sort of, this is the focus of my company, not viruses, but knowledge implementation and dissemination. And then I'll get into the Python. So I've got Python after I tell you about what we're doing and why we're doing it. So hopefully that's a good flow. So my company is Austin Capital Data. We've been around since 2016, and we're very interested in sort of complex deep science domains, but we're interested in anything that has some level of complexity that we can use data science and data visualization to disseminate information about. So the challenge is, in our current ecosphere, our current data ecosphere, there's many, and there's some new ones actually since I wrote this slide. But I mean, data management is an obvious one. Cleaning and organizing data, maybe this stuff is getting worked out as it becomes commoditized. But I think it's still an issue. Analysis and deriving understanding and insight from clean data is always a difficult place, right? Especially if you're working in a domain that you don't understand. Or you're not an expert at, or maybe that's very voluminous. That's also an issue. And then one of our major tools is the knowledge graph. So we're using knowledge graphs to organize these domain specific data sets. So I'm a biologist, so a lot of our work centers around biology, but we don't just limit it to that. And if you have any questions or you want to say anything, just jump in. So the vision is that we have the mission of enabling with data, enabling people to make good decisions and get to data that's out there, because there's this incredible trove of wealth out there. But the technological barriers, the barriers of education, those are all in between people actually deriving benefit. What we want to do is leverage data science against people's biggest problems. Let's see. Data science has changed the landscape for corporations and governments. We know that, but it should also change the landscape for regular humans. So we're seeking ways that we can empower people and increase their ability to do good stuff with the data that belongs to everybody. So Python and Vatical are two of our main tools. So we use knowledge graphs and Python to enable those knowledge graphs. And that's what we'll talk about. We'll talk about using that to understand the viral landscape today. Right. Some background noise. I think there's somebody that needs to mute. Yeah, it's just like somebody eating dinner or something.
I think it's Jacob. I think we can, yeah, somebody.
Cool. It's beautiful now. It's beautifully and clean. I didn't used to have this slide here, but I think this, and I used to be sort of an evangelist for like just basically open data, democratization of data. But recently I've started to question that. There's this common meme here. Do you remember before the internet that it was thought that the cause of collective stupidity was the lack of access to information? It's not, right? We've got plenty of information. We can get plenty of facts. People become arguably more stupid in recent years. I know that's probably not a nice thing to say, but I think there's a downside to data democratization, right? That just saying that pushing information out there is going to solve problems. I think it's a little naive. There's this example of. It's Jeffrey Burdo. He's one of a very high-ranking Catholic priest who was stripped of his. Well, he resigned actually after his data was published in a Catholic newsletter, and so basically his cell phone, collected information about what he was doing, and the data vendor bought that information and sold it to a Catholic newsletter, and they found out where they basically de-anonymized it, right? And then they found out where he was and what he was doing on his off time. So they found out that he was, attending certain bars, and they didn't like that. So the point of bringing that up is that by enabling people to do more powerful work, right, they'll do whatever they think is important to them, right? So this is another thing that's really important. And then there's this thing about free speech in the age of, it's a New York Times article, free speech in the age of disinformation. This is actual information that people have used. There's also disinformation, right? So all these things sort of come into play when we talk about something like, let's pull back the barriers to understanding viruses, right? And understanding, let's dump this stuff in the public space, right? Talk about which viruses are deadly, which ones are not. I mean, there's all sorts of situations that could be positive or negative that have come out of that. So those are the reasons why I bring that stuff up, because we're probably all, technically savvy people working in these domains, and we're in a very different world than we were even a year ago, right? So let's move on. So I believe that now, I believe and I've always believed that building these things into stories, building these things into usable tools that highlight the good and bad aspects of this data sets is a good way to go. So we've built out a number of these things, and we're trying to, the idea behind most of these is to move them to the public domain and give them out as, ways to leverage this information. These are them. So BioPredictor was our first project. The idea was I was actually a newly minted PhD or postdoc actually, and I was looking for jobs. I wanted to know which companies would be the most reliable places that I should go. What would be, I had no idea, which biotech companies would be good ones. So we built a data science project to look at, which ones were more likely to IPO. So that's BioPredictor. Collaboration, another thing that's a constant problem, I think, in science is looking at collaboration, right? So maybe you value a scientist based on the number of publications that they have, but what about their friends? What about the things that they know? What about the things that their friends know? There's a lot more information inside the inside like PubMed, for example, that we can pull out. One collaboration network is one of them. Cancergov, I'm not going to talk about that. I talked about this last time, but I'm not trying to cure cancer. I know Jacob keeps on saying I'm going to cure cancer. That's a huge, that's a huge accusation. What I really want to do is make cancer data useful, open to people who have cancer. Right? Let's say you have cancer. You don't go to the guy down the block. Oh, Jacob, don't go to the guy down the block and ask him what kind of drug should I take to alleviate my symptoms. Maybe you should actually look at the data, right? I look at the PubMed data. So that's Cancergraph. Cocktails is a fun project that we've been working on with a food company to look at cocktail data. And then we come to education. Homeschooling is a big deal right now. How do you judge curriculums? And finally, VirusGraph, which we'll show you. So I'm telling you a lot of background. I'll try and get through it quickly so we can get to the actual Python. But I think it does make sense to argue for this stuff because, like for example, I don't think Trump's a shining example of what you should do, but he was successful at dismantling our pandemic panel, right? So that means that president was able to come into office and say that pandemics are not really a threat, right? So we can dismantle this panel. So, and I think that that's actually the appreciation that pandemics and that viruses are a threat is it needs to be surfaced, right? Even in the age of COVID, because I think people even think that maybe it's over. So these are the major pandemics, right? And this is actually not accurate anymore. What you'll notice here is a number of these things. Actually, I think all of them have vectors, animal vectors, right? And then there's a lot of deaths associated with each of these pandemics. They're probably here to stay. They're probably going to come out. There are probably going to be more and more of these coming around. Thus, my argument that we all need to be educated in terms of viruses, or we need to gear up and understand this. So this was May 2020, right? So that's actually not that long ago. 300,000. We're up 4.5 million deaths right now with COVID from May 2020, right? 300,000. So this is serious, right? And recurrent, and it's not something we have a really good handle on. Let's see. New viral threats are likely to emerge. The 7 major pandemics involve contact with animal intermediate hosts. The reason why this is probably going to happen more is as climate change increases. We've seen wildfires increase. We've seen flooding increase, hurricanes increasing. All these things flush out and populations of animals from places that they weren't there before, and then changing land use patterns, right? And climate change itself. This is going to lead to new contact with wild populations and novel pathogens, novel viruses, right? If these are novel viruses, if these are populations we haven't seen, there's no immunity to them, and there's no vaccine, right? So this is the problem. You can even look at smallpox, measles, and the flus in the Americas. This is coming from guns, germs, and steel, PBS, right? And we know that 90% of Native Americans were killed by these pathogens because they didn't have experience with them, like physiologically. That's my pitch. So what do we do? Well, we can obviously study this. There's a long road ahead. We do have tools to make vaccines, but this is going to require an educated and aggressive population that understands and makes good decisions, right? So that's my pitch for whose knowledge, right? We can leave this in the hands of like the genius doctors, as we were talking about in the beginning. But maybe those experts are not the ones who need to be the only people informed. Maybe we have some sort of crowdsourcing pitch here. Maybe we all need to understand viruses better, right? Maybe the age where we have to understand science as basic literacy is come. So how does that lead to Python, right?
Whoa. I have to stop you.
Yeah.
You showed the picture of a crowd of people.
Yeah.
You haven't shown a cluster of computers. Because this is where you're heading.
Okay.
Now you should, next time you show this, put a cluster of computers, because this is where we're heading.
Well, I like that, but I don't like the Wizard of Oz aspect of it.
I mean, put the Wizard of Oz.
I think, I believe that the people have to be educated as well. It's not just one person driving. Well, this is just an opinion, though, right? This is totally my opinion. But yes, I appreciate that. I think a cluster of computers is certainly a robot army is really
yeah.
Well.
Now you're talking.
yeah. From a computational biologist.
Yeah.
Yeah. Very cool. All right, so let me get into the Python because I notice my time is like ticking away. But yeah, thanks. And keep on telling me what you think. I love to hear it. It's great. So this is a rendering of just the COVID relatives and the proteins that they associate with. So what I'm going to pitch here is that we've got these resources that we can pull in. They're public resources. There's tons of this information. And we can actually analyze it in a principled way, and in a way that I'm going to go back to storytelling, a way that actually is sticky in terms of like a story, right? To get people excited, because you can't, it's not working to just dump, terabytes of data in the public space and then have, only experts acknowledging it. That's my opinion again. But anyway, so this is BioGraken. This is our starting point. This is an open source project, a part of what used to be Graken. Now it's called TypeDB, and they've compiled all these different data sources, some for precision medicine, naming conventions, PubMed, which is 33 million research reports, disease networks, all these things like UniProt or protein networks, DisGenNet. HPA tissues, TissueNet, all these things have to do, this is all the public medical data sources. So we built it on top of that. It had, in addition, it had COVID data. So this was donated by Oxford Pharmagenesis, and they donated information on 19 COVID relatives and 130 interacting proteins. So here's where it gets interesting, I think. So we can take this. You can see the coordinator is the named entity recognition. So there's named entities that are pulled out of those papers. There's also the coronaviruses database. Those are the two pieces that that was added to BioCovid or BioGraken. And then this is, again, you can pull this down. You can run this on your server, and you can look at these interactions. What we want to do though is this, there's such a COVID focus, right? But I think it's really important to think about viruses as a collection of entities that are, beyond COVID, because who knows if coronaviruses are the next plague, right? And maybe it's just a different variant. Maybe it's something that's a little bit different. So Sandeep, who's a data scientist at my company, has been pulling in the Viper virus pathogen resource data, which has over 1.5 million different strains cataloged, right? So there's that, and then we're looking at the string viruses as well. So string viruses has both computationally generated, experimentally generated, and generated by homology interactions between proteins and viruses, right? So it's got viral proteins that interact with human proteins and other host proteins. So all of that. So this is what we're building. You can see Viper, you can see all the different families. Here's coronavirus, but you can see there's 18 other families. So herpes virus, you might recognize some of these virus types, but there's quite a bit of diversity, right? Which I'm going to argue is a vulnerability surface, right? There's a lot of ways that these things can attack. They're very diverse. Okay, so yeah. So string viruses has intervirus and virus host, right? So virus and virus host curated data, experimentally data, and then automatically generated text mine data, and then transfer data, which is the orthologs. You can read about it in this paper if you want. This is our workflow. So basically we're taking our databases. We use the Python client for TypeDB, stuff it into TypeDB, pull out metapaths, right? So we're pulling out paths, virus, protein, virus, for example. And then we're visualizing those shared paths, right? That's the main workflow. So how do you do this computationally? First, you have to build a Grackle file to update the biographing schema, download the data or use an API, build a loader for each data source, insert the data, query the data, and then visualize, right? We're using PyVis, holoviews, and data shader for that. Okay, so here's the code structure. You can see that we have this folder structure. The migrator is the main entry point, but the migrators are each database has its own migrator. That's how we built it out, copying the Gracken pattern. And then the data sets are loaded. We usually download the thing as like a TSV or CSV. You can see that over here, and then we just parse it and stuff it into the database. So that's that. You can see the schema. The schema is very rich. I think one of the strengths of this tool is the schema is very rich. You've got entities and sub-entities. So organism is a sub-entity of thing. Virus is a sub-entity of organism, so we can string them together in that inheritance structure. And then we've got things like attributes. So this one owns its own name. It also has roles, right? So this organism plays an organism association, and it also plays an organism virus host, right? So it can do those things. Yeah.
Is this extracted, or is it something that you give as input?
You give this as an input. It's like an SQL schema, but it's a GQL schema for a graph. So it's just a text that you throw in there as an input. So yeah. So I guess entity, attributes, and roles, that's those two things. Then we've also got relations and rules. So you can have like a discovery as a relation, relating discovered virus to discovered location, discovering location. And then you can have a rule, which is the inference engine. So you can make a rule. This is a simple rule. It's like virus gene, then virus protein. So if there's no translation between gene and protein, we can use the rule to make the translation, right? To make that connection, but it can get more elaborate. Okay, so I'll just try and go fast because I know we're running out low on time, but I want to show you my results.
Take your time. We're not rushing you.
Okay. Probably like a few more minutes then, and we'll get there. I don't want to. Okay, yeah.
Because otherwise, I'll be asking you many questions.
Okay. All right. So I'm gonna take my time. So we import each of these migrators individually. You can see all the different imports. That's the first step. Second step is set the parameters. So you can see that we've got our, localhost 8000. You've got a specific database. We'll layer these on. We've got all our maximum numbers of items. The next step is the migrator. So the migrator really just calls each of these with the URI connection and the database, right? So it's just like very much like a SQL load, right? So it does each of these in turn, loading them up, and then timer, and tells you how long everything works. So that's the DB migrator. We can take a look at the virus migrator internally just to see what it is. So we've got CSV. We're going to import the client, the Kraken client, OS. You can see the definition for the general migrator, and it's very similar to SQL again, like a database, any database. You've got the client, right? Load that as client. The client is going to open a session, and each session opens a transaction. So there's those three levels of nested objects you have to instantiate, and then the transaction type. Each transaction is a read or a write. Yeah. So it all should be very familiar from your database engineering. Let's see. Then here's our parsing code, right? So you can see it's like flying by me. Again, I told you we basically download these all the data frames if they're large, if they're small enough, into the local, and then parse them one piece by piece, right? So you can open your file, open your transaction, and then go from row to row, and then we're going to read in each of the columns, right? Each of the item from each of the columns as a different data item. Yeah, you can see it's a dictionary there. So that's a data dictionary. That dictionary will be sent in as a insert statement. So here's the Grackle statement. It's just wrapped in Python. So for Q in import file, right? So we do this for each one of them. You can see what are we using here? F strings, probably the best way to do this. So basically, you parse out the variable into each of these slots in the F string, like the virus name, and then here's the Grackle insert statement. So you match variable C as country, has country name, so we would change the country name. Insert V as a virus, right? So that's a variable V, it's stuffed there. Has gene bank ID, so we're changing the different ID for that virus. Has virus name, you can see the strain name going in, and then is an organism, has organism name, right? And then we've got this relation. This is a relation right here, discovering location and discovering virus, right? And you've got you were populating it by the variables C and V, country and virus. And then there's the type of relation is a discovery. So that relation relates these two roles, right? Location and virus. And then the second relation is hosting organism and hosted virus. So hopefully, as I'm saying this, you can, in their mind, see how the graph is coming together just by being populated just by these relations and entities that are being stuffed. Blah. And print Grackle, so you print it, and then we insert the thing with an insert call. Again, very similar to any SQL workflow, right? So that's really it. The next thing would be to do is to query the graph, right? And then visualize. And I'm going to jump ahead to just the analysis here. So we query the graph using the same syntax and the same connection structure. And we'll start looking at the objects and the links. So the first step would be looking at we've got data on viruses and proteins. We're interested in perhaps what proteins in the human are attacked by the virus. And we have a hand-curated data set plus that string data set, which is automatic. So we can say this virus attacks or is likely to attack this protein. You can do this analysis. This is a very typical like capture type of bioinformatics analysis. We've got a virus or viral protein. You want to see what sticks to it. You wash a bunch of human DNA or human proteins over it and see what sticks. And then you can get enrichment, right? This leads to what they call Goterm analysis, which is a very standard way to gene ontology analysis. But what you get out is sort of blah. It's like 50% these are cellular nitrogen compound metabolic processes. 50% are biosynthetic processes. 50% are transport. Obviously, these things overlap. and 47% are signal transduction. Okay, that's like not really that informative, right? Like it's not very sticky. This is the problem with the high level of technology, right? These, you've really got to get in and do the experimental science if you want to get anything out of this, right? What does this mean? It's like we've got 30 bolts in a car, we've got some rubber and some leather. Who cares, right? So what I did was I looked at the enrichment of these terms. So we've got 7 viruses with protein data, 99 proteins with private connections. That means that like of all these viruses, only one virus touched the protein, but there are 7 proteins with 2 or more connections, right? So actually, I think that's what that means there. So these blue ones are actually human proteins that are hit by more than one virus, right? So this means that these things might be interesting because the virus is like a common attack surface structure, right? So let's move forward and we can look at these things. ROA2 and ZCRB1 are hit by 3 of those 7, right? So that's actually really common. And then there's ACE2, BTF3, EIF3F, FGL2, and ROA1. If you're a biologist, these things may make some sense. It may not make some sense otherwise. Some of these things are elongation factor like this over here. Some of them are zinc finger proteins. These are things that direct transcription and programs in the cell, right? So there's a lot of maybe signal that sort of leads to transcription and controlling gene expression. So let's dig a little deeper. And what I decided to do was actually traverse the graph further. Actually, this is really what you saw, where we've got virus 1, protein, virus 2. So 2 viruses that are interacting through a common protein. That's the so you can see the query right here. Match V1 is a virus, P1 is a protein. We've got a relation where hosting virus, associated virus is a protein virus association. And then V2 is a virus, hosting protein, associated virus is a protein virus association. So you can see it's the same relation with 2 different viruses, right? V1 and V2. And you can see this here with this familiar SARS-CoV-2, all the proteins that this interacts with, but these 2 are shared by these other 2 relatives. So these are the ones that we're interested in. And that's where the graph really makes this story sort of come together, where we can start to think about you know, what are these common pathways? How easy is it to get to from one to the other? How many viruses are linked to this single one? How many steps is it to get to a third virus? Those kind of questions. Let's see. So we can just look at this, FGL2 and ACE2. So another way to look at this is using data shader and holoviews. So you can see the graph over here. If I zoom in, let's just zoom in on this. You can see the blue nodes are your viruses, and this is a private protein that's only this virus works with. That's ACDB5, which is a fat binding protein. But there are other ones, like if I click on that, I can see that, like for example, SARS-CoV here interacts indirectly with many other proteins in this network. Those are the red nodes. You can see blues are the viruses, right? There's one there. And then the red nodes are proteins, and some of them are on the way to another virus, right? And then we can use this Chord diagram to plot the same data. But you can see, like, I think a lot of this is sort of like research bias, but SARS-CoV-2 interacts with a lot of the proteins all around. And we can look at other relatives and see SARS-CoV-2 GZO2 has a little bit less of a signal. MERS-CoV-2, another variant, has even less of a signal, those red lines, right, connecting to other proteins. MHV actually is pretty highly connected. IBV, another relative, you can see again, the lines are getting sparser, right? And then this variant is actually very different. So those are the types of things that I think you can get at thinking about with the graph, right? There's one last piece that I want to point out. If we just we go forward, we can array these, and you can see sort of how these have decreasing interconnectedness in this network, the story is probably like, this is highly studied and this is very similar, right? Something like this. The second part that I want to just talk about briefly is that we can skip over this. This is just some of the biological detail showing that one of the things that we caught is there's actually research data to show that the same type of protein is implicated in HIV infections, or adenovirus, I think, human adenovirus, right? There's that. But the other thing is this: biological viruses are not the only viral threat to humans. We've got very rich data, but I think we might be going about thinking about it wrong. And if you come from a computer science perspective, we know that there are computer viruses as well. And these things attack surfaces, they attack specific vulnerabilities in the computer, very analogous to the protein stuff that I just showed you. So just very quickly, I want to introduce 621. There's an ontology that you can think about things as like different parts of the computer that are attacked by a virus, like a tool. That's legitimate software that can be used by threat actors to perform attacks. And that could be like, your transcription machinery. Vulnerability, mistaken software that can be directly used by a hacker to gain access into a system or network. Similarly, the body's got vulnerabilities and tools, right? So I just wanted to introduce this because this is sort of the punchline, right? That computer scientists have something to give back. If we think about the story as a threat surface, if we think about the story as like responding to a specific attack, and we can build this out just a little bit more. This actually has quite a bit of richness in the STIX 2.1 set of objects. So that's the talk. Hopefully that wasn't too fast. Hopefully that was interesting enough. Oh, and I should thank my team. So let me just get to that. So obviously Sandeep is working on this actively, but I've got a team here in Austin and Berkeley mostly, who are always working on these projects with me. So.
All right, thank you. Yeah, so that's awesome. I do have a question. So like on the virus side, like what kind of actions can you take based on that data? Like what kind of, like you look at these things and then what can you do about them, or what would somebody do with them, for example, with this kind of data?
Yeah, sure. I mean, like a virus, needs to usually bind through some sort of gateway protein, right? So if there's a protein that a lot of viruses are using to get into a cell, like a transporter, it would be useful to know, because we could either inactivate that, in some way, or maybe block that transporter. I'm not sure exactly what the biology would be, but I mean, like you can think about like COVID, right? We knew about the spike protein, or Moderna did, and they knocked down the spike protein, so they knocked that piece off of the virus essentially, and inhibited it from getting into the cell, right? So like very mechanistically, if we understand the pieces that the virus needs to attack, like again, we can. I love this analogy. We can think about like if you're using some copy functionality right inside a computer, right? If you inactivate that copy functionality, then a computer virus might stop replicating a specific one, right? We can do the same thing, knock down the transcription factor that was actually used, in the adenovirus case, and you can see, just harp back to that because it's such a cool example. You can see in this case what they've done is they've taken, this is a knockout, right, of the zinc finger protein. It's very similar to the one we found. And you can see that the viral proteins are not constructed at the same rate, right? And if you have the wild type or the normal one, the viral proteins are constructed at the same rate. So this could be a target of a drug, like quite literally.
Gunnar, can I ask a question? Are you done with that one?
Mm-hmm.
So I used to study biochem actually a long time ago, but I was wondering on how are you modeling? So I remember like the tertiary and quaternary structures play the biggest role. So there's the primary, and then anyway, the tertiary, the folding structure of the proteins play the biggest role in how they interact with each other. How have you done models, and how do you do a model based on just these kind of genome sequences to know like why let's say the COVID and the COVID Delta, why is one more virulent than the other? How do you do that?
Yeah.
Coded format. How do you take in consideration the three-dimensional structure in code?
Right. So, I mean, the data that I'm approaching right now it's at a much higher granularity, much lower granularity than the folding structure. It would just identify the actors, and it would be, so it wouldn't talk about how the protein folded, but it would just talk about what are the different elements that would be important. But I do know that there's computational databases that are predicting protein folding. I think there's one that just was published that's very accurate. And it's not my domain of study, but we could certainly, if this project gets built out more, think about like much more fine-grained analysis of folding and folding variants. But it's just that's so far from where we're with the stuff I'm presenting now, which is just essentially identifying objects, like biological objects.
Gotcha. Thank you so much. It's fascinating stuff. Thank you.
Yeah, no problem.
I'm looking for a scoop. Yeah, go ahead.
Sorry, I was going to say we have a question in the chat from Jeff Pape, and he's asking, would this technology also be useful for analyzing prions for things like mad cow disease?
So prions, I think, cause protein misfolding. Again, quite far from my domain of expertise, but I mean, I've read a little bit about it. So I don't know. I mean, I think, as I understand it, prions cause like misfolding in other proteins, right? So the prion itself has some misfolding, and that sets off some sort of cascade. So it's kind of an interesting question, which I've never thought about. But I wonder if there would be some collection of proteins that are more vulnerable than other ones. Maybe that could be put in there. That's interesting. But yeah. Maybe. I'm not sure.
Awesome. Thank you for that answer.
Okay, I'm looking for a scoop. And the question is, can you give me one? And then I'll be slow here on. So when I show new technologies to people, they look at it and say, yeah, but can you do this certain thing that you'd say for certainty that this is correct? And usually I tell them things like, look, I can say X or Y, but it never convinces them. It's like they have to have this something that is supported by some higher power over there. So now the question is, from all the analysis of COVID that you showed us, you showed some proteins, and you can say, okay, this is probably more, it's just certain proteins connect to multiple viruses, maybe it's better to look at it. Can you give us some scoop from all the work you've done so far? I don't have to, you don't have to sign on it or anything, but something that is people to remember by this one thing we discovered that it's like seems very important.
Well, okay, in terms of discovery, it's this, right? So it's the fact that this zinc finger ZCRB1, right? Zinc finger C, it's a very specific CCHC type RNA binding motif protein, right? Which has got a very, this U11, U12 small ribonuclear protein, right? So it's got these very specific domains, right? So when I look that up, and there's these other two ones, and this one has, amongst this very small collection of viruses, right? Let's see if I can find it. It's got three of the viruses all hit this exact same protein. They're all working with this same protein from the hand curation that somebody did. And then when you look this up, it turns out that there's, in a different type of viral biology, right? This would be CCH, zinc finger type proteins. I think that might be a misspelling. But anyway, it's also zinc finger type protein with a similar motif, and it it acts in human adenovirus infections. And this is backed up by an experimental study, right? So that's the surprising part, I think, is that by doing this bioinformatic analysis, usually you can maybe speculate, give some confidence, but this actually bears out that there's somebody who's found something when they knock this out, that actually that virus can't replicate as much. And it makes sense because we know that that type of that protein is causing transcription of these late mRNAs, right? So it's actually the pieces that the virus uses to assemble itself, right? So maybe it's not an.
Let me interpret it to people to my level, which I don't understand half the stuff you're showing me, but I'll try to translate and you'll correct me. So you're telling me that looking at all of those data sources and combining them using your knowledge graph is pointed out to a specific protein, and that protein, if you study it in the future, you might find some sort of solution because multiple viruses are looking at it. And you're telling me that someone already has looked at it and said there is something with this protein.
Yeah, yeah.
So, and tomorrow you can do this for many other proteins or many other problems. And you even mentioned like computer viruses. So you can repeat the same process to many, many types of knowledges.
Yeah. We've got like 7,000 viruses loaded now. So, and we're putting in the protein interaction. So we'll be able to get an entire, universe of viruses.
Okay, and just to explain how hard or easy it is compared to doing this without this technology. Without this technology, how many years would people have to study to discover the same thing? How many experiments they'll have to do? But basically, I'm trying to say how much cost this technology will reduce compared to the human endeavor of actually doing experiments the old way in wet labs.
Yeah, and I don't think, in biology without an experiment, nobody's going to believe it, So I can't say it's either or, but the fact that I can point out that there's a study that's already done is a huge cost savings, right? So then
how huge? We're talking about thousands times more, like compared to the cost of a computer and a bunch of data scientists.
Oh yeah, I mean, like if you could, we put our finger on this, and somebody's probably spent 3 to 5 years studying this, right, on an NIH grant. So I would say that if we pull out more data, more studies, right, we don't have to do those, and then we can do this across all 20,000 proteins and all 7,000 viruses, and find that's where the sticks computer virus stuff comes in. We can find those sensitive parts of the cell, right? Those parts, and then identify them and see if we can get a signal that way. So yeah, I'd say I can't even put a price on that because first of all, to master all that knowledge, you'd need to read all those papers, right?
And how much time will it take to read the paper compared to the machine? Just to give people example.
Oh, I don't know. There's 33 million papers inside. You can't do that, right? Inside PubMed, there's 33 million papers. I mean, obviously, if you're looking for something, you can hunt it down. But even then, like when you start asking about adenovirus or HIV, you're probably dealing with millions of papers or hundreds of thousands, right? It's an impossible task.
So basically what you're saying is that this technology or technologies similar to the ones that you are developing here or using now, will accelerate science dramatically.
Yeah. Oh, yeah.
Here's my scoop. Thank you.
Okay. Fantastic. Thank you, Jacob. I've never heard it put so concisely and powerfully.
Any more questions?
I guess I scared the ball.
No, yeah. I remember seeing one time. I was thinking about your study and everything. I can see how I think I saw somewhere where they're doing protein kinetic studies, and they're aggregating the outputs into the data sets. So like you have all these little assays, and so the computer's telling you the percentages of dilutions, let's say, over all the assays, and then what it does is that the data will be aggregated from some kind of analysis tool, and then it puts it into the database. And then what it does is that with your kind of study, I'm thinking you could do modeling in terms of how the different dilution affects the kinetics of the protein. That's what I was thinking. But I mean, yeah, yours is very linear. That's what I was thinking. But in kinetics, you can do it linearly too. You can get data that's pretty linear. But am I off? I don't know. I haven't done this in a long time, but it's very interesting.
I think, you know, like the stuff I'm presenting now, again, is very broad stroke, right? The type of study that you're talking about is much more of like a systematic, high throughput experiment type of study. There's a lot of databases like that with you know, gigabytes of data, right? But the problem, the real pain point is not that the data is not generated or that there's not, you know, systematic study. It's that nobody can get at it and understand it. Right? That's really the pain point. Is that people just don't get it and understand it. It's really like a data visualization problem, in my estimation.
Thank you for the elaboration.
Yes, thank you for your presentation. I was wondering how we could work with cancer.gov. Emailed you as well, and using NIH BioWar, because that's where there's copious amounts of libraries and datas, but it is innovating ways for teams to process the data and translate the data with all the medical artists and all the engineers, because that's where the knowledge gap and skills gap and why you have an assistant to a surgeon, a second pair of eyes, because sometimes that's how unbelievably they leave some scalpel or some material inside, you know, and it's like, hey, stop thinking about the golf game, focus now. And you actually have a second assistant in there in the room, but you know, you need these things. The same thing on a captain on a ship, you know, second set of eyes or five eyes even to help guide.
Yeah, I really appreciate that. Yeah, maybe a whole room full of eyes and robots.
Absolutely, yes.
David, Gunnar gave a talk about how he used this technology for cancer. If you look up at past Python meetups last year. So
oh, excellent. Okay.
Yeah, so he's already going in the cancer direction. He just today he's talked about more about COVID and viruses, but this technology can be applied to multiple things, as showed.
Let me give a pitch for my talk in a week. I'm going to talk about Cancergraph and all things open. It's free to register for. So
please do.
Yeah, I'll throw that right in
Thank
chat.
you.
Yeah.
Yes.
Cool. Yeah, please come to that. That's great.
Will do. Thank you so much.
All right, more questions? Going once, going twice. All right, Gunnar, thank you so much. This was a very interesting talk and very impactful to everyday life. And I always enjoy watching, like, seeing these talks on biology, because they always have a real application, a real impact for everybody in the world. So, yeah, thank you for doing this, and thank you again for coming to our meetup and showing this to us. Thanks so much.
Thank you. Appreciate it. Thanks a lot.
All right.